Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on gene mutation. Previously, we discussed the process as well as the site of translation within cells. We learned how proteins are made through translation. We also discussed that in order for our bodies to synthesize protein, we require a continuous supply of amino acids from the food that we eat. In today's lesson, we will describe gene mutation. We will also learn about missense, nonsense, and frameshift mutations. A mutation is any spontaneous change in the genetic material of an organism. These changes range from large structural transfigurations that involve part or whole chromosomes to small alterations involving just a single base. It is the small mutations, such as those involving a single base, that cause the most concern. These are referred to as point mutations. There are a few different types of point mutations, including substitution, deletion, and addition. These mutations, which each involve a change to one base, occur unpredictably when DNA is replicating. The changes that these mutations cause in a gene can be quite substantial. At times, a gene may code an incorrect protein, or even no protein at all. Let us now discuss the three different types of point mutation. In substitution mutations, one base is replaced by different bases, as you can see here. In this example, guanine has replaced thymine, and the triplet ATT has been changed to ATG. While the original triplet codes for isoleucine, which is an amino acid, the new triplet codes for methionine. This substitution results in a different protein being synthesized. However, it is important to note that one different amino acid in a protein will not always create a functional change. In our example, if the substitution had been any base other than guanine, because the DNA code is degenerate, the triplet would have still coded for isoleucine. Thus, the same protein still would have been synthesized, and the gene would have been the same. Other substitution results in a stop triplet, as you see here. when the transcription reaches the stop code, it halts. This results in a non-functional mRNA. This is also known as a nonsense substitution. In deletion mutation, a base is missed out during replication. In addition mutation, an extra base is added. Both of these mutations are more consequential than substitutions because they can never result in the production of the same protein. This is because they do not change the triplet in which the mutation occurs. Rather, they remove from it or add to it, altering the entire sequence. A frameshift has occurred, and thus we refer to these types of mutations as frameshift mutations. These mutations result in the production of a completely different mRNA, if one is produced at all, and a non-functional protein, if one is produced at all. In some cases, a frameshift can result in the deletion or addition of an entire triplet. This results in either one extra or one fewer codon within the mRNA. This, in turn, will lead to one extra or one fewer amino acid in the polypeptide. Students, let us do an activity. What are the 3 types of point mutations that we have discussed? Please describe what causes each mutation and what happens as a result.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. I am sure that you had no trouble completing this activity. The first type of point mutation we discussed was substitution. This occurs when one base is replaced by a different base. Substitution may or may not result in a different protein being synthesized, and the new protein may or may not be significantly different from the original. Thus, substitution does not always result in a functional change. The second point mutation we discussed was deletion. This occurs when a base is missed out during replication. The third point mutation is addition, which occurs when an extra base is added. These mutations are known as frameshift mutations. Frameshift mutations always result in a new protein being synthesized. They result in the production of a completely different mRNA, if one is produced at all, and a non-functional protein, if produced at all. Were your answers correct? If so, you're doing well. Let us discuss a fun way that you can think about frameshifts. This will help you remember how they function. Students, look at this sequence of letters. If we give this a reading frame of 3 letters, similar to how triplets are arranged, we end up with this sentence. The man was hot and ran for his hat. That makes sense. However, if we take out the S at the end of was, representing a deletion mutation, we end up with this. Our sentence no longer makes any sense, does it? In generic terms, it is missense coding. Students, please complete the following statements.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello again, students. I hope you did not find this activity too difficult. A mutation is any spontaneous change in the genetic material of an organism. Point mutations occur randomly when DNA replicates. When transcription reaches a stop code, it ceases, and a non-functional mRNA results. When a whole triplet is missed or inserted, it results in either one extra or one fewer codon in the mRNA. Today, we discussed gene mutations, including missense, nonsense, and frameshift mutations. In our next lesson, we will learn about chromosomal mutations. This brings us to the end of our lesson for today. Until next time, thank you, teacher. Thank you, students.